Hello. So, for the last part of this lecture, I think what I'd like to do is to introduce you to three common kinds of experiments that you might meet in the literature when you're reading about ecology and evolution. And they are three examples of ways in which we can particularly clever or creative ways in which researchers use messy biological data in order to isolate and capture the effects of a particular manipulation or particular variable of interest. And they are, first I'm going to go through something called a common garden experiment, then we're going to go through something called a natural experiment, and then we're going to go through something called artificial selection experiments. So a common garden experiment, which is also known as a transplant experiment, is a really nice way of asking the question, is a particular difference, or is a particular trait difference down to genetics, or is it mostly down to environment? Is a difference you see in nature predominantly genetic or predominantly environmental? And what you do with a common garden experiment is that you take individuals displaying particular differences in either different environments or different locations, and you bring them into a common environment so that you raise them, or you raise them, or you raise their offspring in identical conditions, basically. So a good example of how to ask this is a study by Field et al., where what Jeremy Field wanted to do was to ask in across Britain, there's a bee called Halictus rubicundus, and that shows social behaviour in the south of England and solitary behaviour in the north of England. And what Field et al. Wanted to know was whether these represent different different races of bees, whether they are genetically separate, or whether they are responding to different environments in the north and the south, and whether they would display different behaviour if you moved them from north to south. And so they did a transplant experiment. So they took colonies from the north, or they took breeding females from the north and brought them to the south, and then they did the reverse experiment where they took females from the south and brought them to the north. And then they observed what the resulting offspring would do. So obviously, if the transplanted individuals behaved the same as they would have done in their home location, if the southern ones ended up all social in their new northern location, and if the northern ones ended up being solitary in their new southern location, then you could conclude that the difference was genetic. But if they all display the patterns characteristic of their new location, then if anything moved to the north becomes solitary and anything moved to the south becomes social, then you can conclude that the difference is mostly environmental. And what they found was that individuals moved to the north, their offspring were all solitary, and individuals moved to the south, regardless of origin, were all social. And so the answer to that question is very nicely that they're mainly responding to environmental cues. And Field et al. Supposed that was because of differences in day length and season length between the north and the south of England. There's more time to pack in two generations of bees in the south, and so you can get away with being a social bee down in the south of England. So very, very nice transplant experiment called another name for it being a common garden experiment. Then we come to natural experiments. Natural experiments are where you take advantage of an opportunity provided by a natural event in retrospect. So one really cool example of this, and you look at differences before and after the event. So a really cool example of this is, we all know the example of the peppered moths, where you can see in the picture, you have two forms of moth in in England and Kettlewell in the 50s found that the black form of the moth got protection from predators when sitting on black trees, and then the trees were all black around industrial Manchester and Liverpool, whereas the white form was quickly picked off by predators because it was conspicuous. And then when you went into rural areas, the reverse happened, because the trees are there covered in lichen, and the white form is more protected by being camouflaged against the lichen, and the black form suffers a lot of predation. And in subsequent years, it was found that, in the 70s, you found that as you go from Wales in the west, all the way across to kind of very, very rural Wales, all the way across to industrial Liverpool and Manchester in the east, you get this cline, this kind of increase in the frequency of the black form, corresponding to the frequency of sooty, blackened trees from the industrial, from all of the smoke that's being pumped out by the heavy industry. And so that was linked to the reproductive success of the black form of the moth. And that kind of pattern is called a genetic cline, where you get a change in the frequency of one allele, and you can link it to the selective pressures involved. But Sakari et al. A few years ago made use of the fact that since 1993 we've had the Clean Air Act, which has outlawed a lot of these industrial practices which pump smoke out into the environment. And the average colour of a tree, even in urban Liverpool and Manchester, the trees are no longer black. And they reasoned that selective pressure would not be quite so strong since the Clean Air Act. And since all the trees had stopped being black, the black form of the moth would likely not get quite such an advantage. And so in 2008, 15 years after the original study, 15 years after the Clean Air Act, Sakari et al. Went back to these locations and measured this genetic cline again. And what they found was that the frequency of the carbonaria form, the black form, is much, much, much less than it used to be. And that has much less of an advantage across that genetic cline, and the change in frequency is much less strong. What they found was they used that natural experiment of the Clean Air Act, kind of changing the colours of the trees, to test the hypothesis that it was originally the selective pressure of the black trees that led to the change in frequency of the black form of moth. And it's strong confirmation of that idea. So that's a natural experiment. And then finally, we have the artificial selection experiments. Artificial selection experiments are where researchers create genetic lineages of experimental animals, and they subject one line to particular kinds of selection and another line to another kind of selection. And over generations, they observe changes. And this can be a very, very powerful way of detecting linkages between traits. For example, Wilkinson carried out a set of very famous experiments on stalk-eyed flies. And they have these wonderful kind of eyes on the end of these kind of crazy, crazy stalks. The reason we think the males have eyes on stalks is because females prefer males with long eye stalks. And according to theory, what should happen is that over time, the male trait of having eyes on stalks should become genetically linked to the female preference, i.e. they occur on the same chromosome. And so what Wilkinson did was to create two lineages of flies with different lengths of eye span, where they selected in one lineage they selected to breed males with really long eye spans, and they just basically, all they did was the only flies they allowed to breed were the males with massive eye spans. And then in another lineage, they chose males to breed which had a shorter eye span. So they didn't do anything with the females. They just chose males to breed which had long eye spans. And they obviously were successful because, as you can see, over time, over generations, the short lines, the male eye span got shorter, and the long lines, the male eye span got longer. But the real question they wanted to ask was, is there a linked female preference? And so after loads and loads of generations, they assessed the preference of the females for longer or shorter eye spans. And what they found was that in 2 unselected lines, females had a preference for longer eye spans, as was originally supposed. And in their long line, the females still had a strong preference for long eye spans. But then in the selection line where they had chosen short eye span males, the female preference had reversed. So females in that lineage had a preference for shorter eye spans, which shows that even if you're just selecting on the males, you are also affecting the female preference, which is nice evidence that those two are genetically linked. And that's a really, really nice example of an artificial selection experiment. And they've been used for all sorts of things to do with quantitative genetics and genetic heritability and that kind of thing. Okay, so those are 3 kinds of classic experimental types in ecology and evolution. And just to recap these last 2 lectures, we've had a lecture on basics and pitfalls, asking why we do experiments, what can commonly go wrong with experiments, and I gave you a checklist for what I want you to think about when you're designing your experiment, especially planning with the analysis in mind. And then in this lecture, we've dealt with the way you can structure your predictor variables, and we've dealt with ways in which you can use your experimental design to minimize confounds, things like randomization and blocking and minimizing pseudo-replication. And then in this final video, we've been through several classic experimental types in ecology and evolution. We've been through common garden experiments or transplant experiments, we've been through natural experiments, and we've been through artificial selection experiments. So I hope you've enjoyed these 2 lectures on experimental design, and I wish you good luck with the rest of the module.